Good morning, everyone. So I'm going to present some of our data on land use associations with some of the PFAS we have been seeing in smallmouth bass in the Chesapeake Bay watershed and some nearby watersheds as well. Next. So since the early 2000s, we have been doing a lot of looking at smallmouth bass health issues as well as population declines in Pennsylvania, West Virginia, Maryland, and Virginia. We've seen fish kills. We've seen a high prevalence of lesions due to multiple viral, bacterial, and parasitic infections, which to us suggests that immunomodulation is occurring. We also see intersex, which indicates exposure to endocrine disruptors. And so, starting in 2013, we conducted comprehensive fish health assessments in those states. And as part of that, fish were bled. We obtained the blood sample, and then back at the lab, we got plasma from those blood samples, and we aliquoted that and stored it at -80 in case we would have things down the road we wanted to look at. At four of those sites, we also did surface water samples, and they were analyzed for a large suite of contaminants, but that included pesticides, 105 different pesticides. They were sampled monthly most of the year, and then bi-monthly in the spring from 2016 through 2019. Next. As PFAS became recognized as a potential issue and potentially contributing to some of the disease issues we were seeing in smallmouth bass, we took some of that archived plasma that we had collected from four sites in 2018. And early on, they were analyzed for 13 different PFAS. Now, as we move on, we're analyzing for more. But four PFAS were found in every single smallmouth bass plasma sample. And as you can see from the graphs below, PFOS had the highest concentrations. And if you look at the bar, PFOS is usually an order of magnitude higher than the other three. But the other thing you see in those graphs is that there was quite a bit of site difference. So Antietam Creek, which is the black bar, had the highest of all of the different compounds. West Branch Macantongo, which is in the Susquehanna drainage, had the second highest. And then the South Branch Potomac near Moorfield and Pine Creek, also in the Susquehanna drainage, had the lowest. Next. And when we looked at potential land use associated with those, what we saw was that, for instance, Antietam Creek had the highest developed land, which is often associated with exposure to PFAS. But it also has a high agricultural, the highest, 49%. West Branch Macantongo, which was the second highest, had the second highest percent of agriculture, and then South Branch, Potomac, and Pine Creek have the lowest agriculture as well as the lowest developed land use. So that did make us think more about agricultural sources for some of these compounds. Next. And this is just a way to kind of look at it graphically. On the right-hand side, you see that graph again for the PFOS and the site differences. And if you look at both agricultural land use and developed land use, you see somewhat of the same pattern, with Antietam having the highest, West Branch Macantongo the second, and then the South Branch Potomac as well as Pine the lowest. Next. So we did decide that all of this data did indicate that PFAS might be having an effect and that we should look at a broader spatial assessment. And so the map on the right gives you an idea of where the sites were that we collected. This includes the same four sites that I mentioned we did in 2018. We also had a site that's out of the Chesapeake Cheat River in West Virginia. Then we added 2 extra sites in the Susquehanna. SS is the main stem Susquehanna near Selinsgrove, and CC is Chillisquaukie Creek. And then the 2 highest on the graph, SC is Swatara Creek, and LNC is Little Neshaminy Creek. Next slide. And again, when we look at the upstream land use for these, Little Neshaminy Creek is actually close to Philadelphia. It's impacted by the Willow Grove military base, and so we expected it would have the highest levels, and indeed it did. It also has the highest level of developed land and a lower agricultural land use. On the right-hand side, EPA has put out a list of potential facilities that potentially release PFAS, and so we also looked at those. So if you look at the 2 that had the highest, Little Neshaminy and Swatara Creek, they do have the highest, and Antietam Creek also has a high number of potential point sources. But our interest was kind of in those middle ones, the West Branch, Macantango, Chillisquaukie, and including Antietam, where you have a high developed land use. And for some of those, particularly West Branch, Macantango, and Chillisquaukie, you don't have many point sources, and developed land is also lower. So again, our interest was, what are the possible sources from agriculture? Next slide. This is just a spatial assessment and a graphical to show you the 4 PFAS that were found in every plasma sample. And what you see, the graph that I had previously was total PFAS. And what you see is that PFOS is often very similar to the total, because it is always the highest level we see in smallmouth bass plasma. And again, even in this spatial distribution, PFOS is usually an order of magnitude higher than the other 3 compounds. And you can see there's a little bit of a difference from site to site for those other 3, but again, in general, or for all of them, Little Neshaminy has the highest level, Swatara Creek next, and then Antietam, and then the 3 sites that are more agricultural associated. Next slide. We also looked at those 4 sites that we did over time. We wanted to see, was there much change over time and from year to year at those sites? And as you can see, there is a little bit of change from year to year. Sometimes it's significant, sometimes it's not. But the same pattern among the sites stays the same. So Antietam Creek has the highest, irrespective of the year. West Branch, Macantango, the next highest, and then South Branch and Pine tend to be similar. Next slide. So what are some of the agricultural sources that we're looking at? Obviously, this conference, we're really focusing on pesticides and PFAS, but I do just want to point out that we are very concerned about biosolids as well, and we do think that is an important source in lands that are primarily agricultural. And as far as pesticides, if you were here at the last session, you heard a lot about the sources within the pesticides. So there are pesticides that have PFAS as active ingredients, also some that have PFAS as inert ingredients, and then there can also be leachate from containers. So a number of different sources in pesticides. Next slide. So according to a recent paper, there are 66 registered PFAS pesticides where PFAS is an active ingredient. And as I mentioned before, we had data in surface water for 105 different pesticides that we had analyzed at our 4 temporal sites. And when I looked through this list, at least 16 of those were considered PFAS pesticides. And so I looked at that data to see, were we finding those at our sites? Most of those 16 were not detected, but 2 were detected frequently at the 2 sites, Antietam and West Branch Macantango, which had the highest PFAS concentrations. And those were fipronil and its metabolites, and that's an insecticide, and then norflurazone, which is an herbicide. Next slide. Fipronil was actually detected in all years throughout the year at Antietam Creek, except for a couple non-detects in early spring in some of the years. It was detected only once in 2018 at West Branch Macantango and also at the South Branch Potomac, although at lower levels, and there were no detects at Pine Creek. The norflurazone was detected in both Antietam Creek and West Branch Macantango, and no detects were found in the South Branch or Pine Creek. Next slide. But there were also some other things I think we need to think about as far as this. There is one pesticide, it's an insecticide, sulfuramide, that's used in a range of products to control ants, roaches, and termites. Its use in the US is actually restricted, but this is one of those that is a good indicator of how metabolism of some of these substances, for instance, the ETFOSA, is metabolized to PFOSA, which then is metabolized to the PFOS. And it has been shown to be a major source of PFOS in biosolids. So even though we didn't detect it in water, and actually it was one we didn't look for, it's an example of things that we need to be thinking about, because obviously we're not looking at every single pesticide that we could look at. The other thing is that we did not find bifenthrin, which is one of the PFAS pesticides, in water, but it was detected in sediment samples, and it was actually one of the few that was detected in young of the year smallmouth bass tissue. So again, we need to think of sources. Next slide, please. And when we think of sources for a fish like smallmouth bass, we have to consider not only the surface water, but sediment and also diet or food web issues. So smallmouth bass, particularly the males, build and guard nests during the spawning season, so they're in association with the sediment, and then young of the year also associated with those bottom sediments. So any of these compounds that are actually ending up more commonly in the sediment than in the water or in the food web can be major contributors as well. Next slide. So in conclusion, we are finding PFAS in smallmouth bass, even at sites that are primarily agriculture and forested land use. We do have evidence that pesticides and potentially biosolids are contributing to these levels, and they could come from both surface water and sediments. And the other thing that I think needs a lot more work is we find PFOS at significantly higher concentrations than the other PFAS. But it is one of what's called a terminal PFAS. And so many of the other PFAS can be metabolized to the PFOS. And those are things we definitely need to be looking more at. Next slide. And so I'd just like to end by saying that this was funded by USGS Ecosystems Mission Area, Environmental Health, and Land Management Programs. We are continuing to look, so all the fish that we have PFAS data for in the plasma, we also have lots of biological endpoints from the cellular to the molecular to the organism level. And Heather Walsh and Cheyenne Smith, who work in the lab, are taking that data, and we're trying very hard to see, are we seeing effects from any of these levels that we find in smallmouth bass? And then lastly, I'd like to thank the many state biologists in all the states that work so closely with us and really allow this work to be done. Next slide. And if there's any questions, I will certainly take them. Thank you.